Good afternoon. Welcome back. Today we're going to talk about genomes and genetics. I want to tell you all about the different kinds of virus genomes, what they encode, and how we can manipulate them in the laboratory to study viruses. And this story begins in the 1950s. Now, many of you may know that for many years, people thought that protein was genetic material of cells because nucleic acid, which had been discovered in the 1800s, was too simple. There are only 4 bases. How could that be genetic material? So it wasn't until 1944, experiment done here at the Rockefeller showed that in bacteria, DNA is the genetic material. Fast forward to the 50s, for viruses, the same is found, that the nucleic acid is the genetic code. And I'm going to show you 2 experiments today, one with a phage that shows that DNA is the genetic material of the phage, and another with tobacco mosaic virus, the plant virus, that shows that RNA can also be genetic material. So here is the famous Hershey-Chase experiment with phage T2. So it was done by Alfred Hershey here and Martha Chase, his assistant, in 1952. And this is the famous kitchen blender experiment where they had a phage, a bacteriophage, and they said, what is the genetic material? Is it the protein or the DNA? So they used radioactive precursors to label either the protein or the DNA, and they would infect cells with those labeled phages. And after letting the phage stick on or absorb, they would put it in a blender to remove all the phage. And then they would let the infection go and ask, where is the radioactivity? So that's the kitchen blender. In fact, there is one of the blenders they used. If you go out to Cold Spring Harbor, they have it in a museum there. So on the top, we label the protein. And when you look at the progeny phages, there's no radioactivity in all the new phages that are produced after reproduction. On the bottom, we have viral DNA labeled with phosphorus, and now you find radioactive DNA in the progeny phage. In other words, the DNA that goes in, some of that also gets incorporated into new phages, but not the protein. Same kind of experiment with tobacco mosaic virus, except this is a genetic experiment. So I would say the previous one is a biochemical experiment. This is a clever genetic one. Here they had 2 different strains, let's call them, of tobacco mosaic virus, a red one and a green one. So the virus is a piece of RNA wrapped in protein. We'll learn more about that next time. And so we have a green and a red one. And the observation is you can purify the RNA and the protein apart from each other and mix them back together, and they will make an infectious virus. So they purified the green and the red RNA and mixed the green RNA with red protein and the red RNA with green protein. So you could just add the protein to the RNA, it reassembles, and then you can infect leaves. And lo and behold, what's the color of the coat is determined by the RNA. So if you have the RNA from a green protein, it makes a green virus particle. It doesn't maintain the color of the coat, or the type of coat is not maintained because it's encoded in the RNA. So I'm using red and green just as an example here. There was another way that they distinguished them. So we have DNA and RNA of viruses is the genetic material. I've told you so far that there's so many viruses out there of different kinds that infect so many different things, but it turns out there's only 7 different kinds of viral genomes. Perfect, the number 7 line. We're going to actually use the subway numbers in this course a couple more times, because you guys are here in New York, so that's an easy way for you to remember. And the rest of your life, when you see the number 7, you're going to remember, oh, that was from virology, 7 viral genomes. I do when I take the number 1 train up here. The number 1 is for something else in this course. Actually, don't go on the number 7 very much. 7 viral genomes, though. We're going to look at that today. And the other fact that's important is that all viruses have to make mRNA that can be recognized by ribosomes in the cell, because no virus encodes ribosomes and translation machinery, all of it anyway. So the RNA, mRNA that are made in virus-infected cells has to be recognized by the host cell ribosomes. All the viruses we know follow this rule. There's no exception. So that can also help guide us to understanding the diversity of viruses. And so David Baltimore in the 1970s took this information and devised what we call the Baltimore Scheme. So that's David Baltimore there. I worked with him at MIT. I was his postdoc in the early 80s. He had won a Nobel Prize for identifying reverse transcriptase, which we will talk about more in a bit. But he thought, what if I put mRNA in the middle and then trace the pathway from all the virus genomes to mRNA? And that's the Baltimore Scheme. And these are the 7 different kinds of viral genome. So for example, viruses with double-stranded DNA, those are group 1. Double-stranded DNA can be transcribed to make mRNA. Transcription is the process of making mRNA from double-stranded DNA. And so the pathway to mRNA is very simple. Double-stranded DNA, right to mRNA. Now, single-stranded DNA, we have some viruses with single-stranded DNA. Those are group 2. Single-stranded DNA cannot be transcribed. It has to be made double-stranded first, and then it makes mRNA. And likely, in the same way, we have these gapped DNAs. They're double-stranded with gaps in them, and you'll see why later on in this course. They can't be transcribed, so they have to be repaired to make double-stranded DNA. So we have those 3 different types of DNA genomes. You can easily trace the path to mRNA. All right, so then the rest of them are RNA genomes. We have double-stranded RNA. Double-stranded RNA has a plus and a minus strand. We'll define those in a moment. One is the mRNA. But in a double strand, the ribosomes can't get at the mRNA. So those genomes have to make mRNA from the double strand. We have minus RNA genomes, the opposite polarity of messenger RNA, so they have to be copied to make mRNA to make protein. We have viruses with plus strand RNA, like SARS-CoV-2. They can be directly translated, but to make more of them, you make a negative strand. And finally, we have plus strand RNA viruses that are copied to DNA, first to a single strand of DNA and then to a double strand. And so those are the 7 kinds of genome there are. There aren't any more. Maybe you could think of some other theoretical ones, but every time a new virus is discovered, it's one of these 7 different classes. Now, I used plus and minus already, but let's define them. The plus strand is the mRNA strand. And it's just plus for a convention. It has nothing to do with polarity or electrical charge or anything like that. So the mRNA that's translated is the plus strand. So the opposite is the minus. And DNA strand that is the same polarity as mRNA would also be the plus strand. So if you're transcribing DNA to make mRNA, the enzyme is actually transcribing the minus strand of the DNA. So plus, and its complement is minus. Now, not all plus RNA is mRNA, as you'll see in this course. Sometimes, depending on the virus, it is not actually translated, even though it could be translated. And for example, the viruses that make a DNA copy of their genome, that plus RNA goes into a cell, but it's not translated. It's copied to DNA first. So you have to be very careful that you don't assume just because it's plus polarity that it's mRNA. All right, so those are our definitions. Now, why I like the Baltimore system, I'm a big fan. So plaque assays I love, and right behind plaque assays, the Baltimore system, because it makes it easy to arrange all these different viruses. All you need to know is the nature of the genome. If I tell you plus RNA, you can immediately tell me how to get to protein from that, because if it's plus RNA, in theory, it could be translated directly. If I say plus DNA, you can tell me, well, it has to be made double-stranded DNA first, because only double-stranded DNA can be transcribed. So that's the beauty of this scheme, is you can get from the genome to mRNA. So there are 7 classes of viral genome, and they're all shown here again. Double-stranded DNA, gapped DNA, single-stranded DNA. Those are the 3 DNA types. And then we have RNA of double-stranded, plus and minus, and plus with a DNA intermediate. Those are all represented on that Baltimore scheme. You know, in the old days, I used to say memorize it, but you don't need to memorize anything anymore. You just need to understand. I'd rather you understood things rather than memorize them. Because in this world, you can always look stuff up. Someone once told me, this is a really good idea. There are two kinds of knowledge. There's the kind you carry in your head, and there's the kind you have to look up, and both are useful. You can figure that out. All right, first quiz. Why is mRNA placed at the center of the Baltimore Scheme? A, because all virus particles contain mRNA. B, there's no reason. C, because all viral genomes are mRNAs. D, because mRNA must be made from all viral genomes. E, because Baltimore studied mRNA. Most of you got D, which is correct. Because the mRNA is put in the middle of the Baltimore Scheme because all virus genomes have to lead to mRNA. None of these other answers are, at least none of you picked B. That's good. There's no reason. It's always a reason, right? And all virus particles do not contain mRNA, and all viral genomes are not mRNAs. For example, a double-stranded DNA virus would not be an mRNA, right? It'd be a double-stranded DNA. Even though there's 7 classes of genome, they can be structurally even more diverse, as you can see here. Just to give you an example, again, you don't need to memorize this, but understand that there can be linear or circular double-stranded DNA genomes, as you can see in the top 2. There can be, even RNA genomes can be circular. They could be in one molecule. They could be segmented, like that one where there are 8 segments. In one virus particle, the genome is divided into 8 segments. It could be single-stranded of plus or minus. They can even be ambisense, which means they contain both plus and minus. Double-stranded, they can have proteins attached. They can have the ends of the nucleic acid covalently linked. So lots of structural diversity in those 7 classes. So what is the function of this diversity? And notice this is not a why question. Why questions in biology are hard to answer, but function is easier. So I say, what's the function of genome? Not why. What's the function of genome diversity? So we have both DNA and RNA-based genomes. So what's the function of that? Well, we think that RNA genomes were the first to appear on Earth many years ago when Earth first cooled and we had chemical reactions going on. We think RNA arose and it was self-replicating RNA. That was called the RNA world. It got pretty advanced, but probably didn't lead to the production of very complicated cells. You needed to have DNA genomes for that. So at some point, probably in the RNA world, protein synthesis arose, and that allowed the development of enzymes that could copy RNA, and one of those enzymes changed and suddenly made DNA from the RNA, and that led to the DNA world where genomes could be really big, because as you will see, probably the biggest RNA is just 100,000 bases or so, where DNA can become millions of bases. So to make a cell, to make a human, you need DNA because you can have more coding capacity. So today, the only RNA genomes on the planet are viral. DNA-based organisms have outcompeted everything, and only some RNA viruses remain, although they're very competitive, those RNA viruses. There are entities called viroids, which we think are relics of the RNA world. We think these are some of the molecules that might have existed back in the RNA world, and we will talk about them towards the end of this course. That explains why the function of having both DNA and RNA, because RNA is from a world that doesn't exist anymore. But the function of linear, circular, segmented, double-stranded, single-stranded, plus or minus, the function of those, we have no idea. They all work. They're all evolutionarily competitive. So we can't say that one has an advantage over any other. The only thing I would say is that if you look at all the viruses that cause disease, the vast majority of them are RNA viruses. And I think that this is because RNA virus mutation rates are higher than DNA virus mutation rates. And we'll talk more about that later. So that gives RNA viruses some advantage, but there are still DNA viruses and they still can compete. I always used to put this up and say memorize it, but at some point in this course, we used to take exams in here and everyone could have a sheet of paper and you could put everyone had the Baltimore Scheme on it. So you should just know this and where to look for it. because if I tell you how do you get from single-stranded DNA to mRNA, you'll be able to figure that out. All right, what kind of information is encoded in a virus genome? Well, obviously, things to do protein synthesis. So giant viruses have some components of the protein synthesis apparatus. No virus encodes a complete translation system, but some have parts, presumably to modulate protein synthesis. Viral genomes encode proteins needed to replicate the genome, to package the genome in a capsid, to regulate the reproduction cycle. And then at the bottom here, modulation of host defenses. When we have viruses now infecting a host like us, every virus has to antagonize the immune response. Otherwise, it won't exist. Every virus has to antagonize host defenses in some way. We'll talk about those in the second part of this course. And also gene products involved in spread from one cell or one host to another. This virus is quite interesting. This is called Tupan virus. And there's a meaning of this, which I forgot. But if you Google it, you will find out exactly what it means. It was named so by the discoverers for a particular region. So someone look it up and shout out what it means, please. Thunder God. It's like a South American thing, right? Yeah. Because this virus encodes, well, only the ribosome is lacking. This was discovered by South American and French collaborators. And the French, you can tell this, only the ribosome is lacking. It's not an English construction, right? There's something different about it. So that's from the French. 20 amino acyl tRNA synthetases, 70 tRNAs, multiple initiation elongation proteins, translation-related genes, the most complete translational apparatus of the virosphere. So that's a word I haven't used yet, virospheres, all the viruses on the planet. So this is amazing, and that's the virus particle there as well. It's beautiful. Of course, they've colored it blue, but it's still nice. And only the ribosome is missing. Doesn't encode a ribosome. Does any virus encode one? Maybe. We haven't discovered it. There are lots of viruses out there we have not discovered. Why you would want to have such a translational apparatus? Well, to modulate protein synthesis in the cell, presumably to favor virus production. And people are doing experiments to look at that. Well, what don't we encode in a genome? That's also telling. There are no genes encoding the complete protein synthesis machinery, as we've just said. No genes encoding proteins involved in membrane biosynthesis. But I'll bet they're out there and we just haven't found them. This list used to be longer years ago in this course. And one by one, I take them off as we find these genes in viral genomes. Standard host chromosomes have centromeres and telomeres. You don't see those typically in virus genomes. And as I say, if I say we don't have them, maybe we'll find them. When we find new giant viruses, 90% of the genes, we have no idea what they encode because they don't look like any protein that we've seen before. So it's hard to know when you have that kind of a protein what it does. So I wanted to show you some of the biggest and the smallest in this cast. These are the largest known viral genomes. They're all DNA virus genomes. And starting from Pandoravirus salinus, 2.4 million base pairs of double-stranded DNA, encoding 2,500 proteins, 90% of which we have no idea what they do. The way you figure out what a protein does is by saying, does it look like any other protein? And you can get ideas that way, but many of these don't look like anything. Now, Haemophilus there on the side, 1.8 million bases. That's a bacterium. That's a bacterium that can live on its own, and it's the smallest bacterial genome. So look at how much bigger this is a virus. It's not even a bacterium. Then we have other Pandoraviruses and getting smaller and smaller. There's Tupan virus, which is quite large. And these viruses, by the way, all have lovely names. So Mimi virus was first discovered in France. Mimi means microbe mimic. And then they started to find a series of related viruses. So they got funny, I guess, and they called them Mama virus and Mumu virus, and there are others as well. So the French, in the naming of their viruses, have a sense of humor. Okay, so these are the biggest. The largest RNA virus genomes don't even come close. The biggest one that we know of is 41,000 bases of plus-stranded, single-stranded RNA. It's from a virus that infects planaria. You probably remember planaria from high school biology, the little creatures that you could cut and they would regenerate. Anyway, there are viruses of them, and that's the biggest RNA virus. And then there's two others. There's a virus of aplesia and a virus of a snake. These, by the way, are all related to coronaviruses, same order of viruses. And SARS-CoV-2 would be about 30,000 bases of RNA, but it's not in the top three, obviously. About the smallest known viral genomes. This is a mix of DNA and RNA. So at the top is actually a viroid. Not sure if this is technically a virus. It doesn't encode for anything. You see no protein coding regions. So whether it's really a virus or not quite sure. But anyway, I put it here because the smallest viroid is only 120 bases in length. Then below it is a satellite RNA. So a satellite RNA is something that requires another virus to help it multiply. So this is 220 bases of single-stranded RNA with no protein coding regions. And then we get into viruses that encode proteins. There's hepatitis delta virus, which encodes one protein, 1,700 bases, and then more and more proteins. And then we get into some very small RNA genomes. This Racaniello virus, by the way, is circovirus. Very small, circular, single-stranded DNA genomes. All of you have these in your blood, and they don't seem to do anything negative. All right, next question is, what information may be encoded in a viral genome? A, gene products that catalyze membrane biosynthesis. B, complete protein synthesis systems. C, centromeres or telomeres. D, enzymes to replicate the viral genome. So which may be encoded in a genome? Most of you got D, enzymes to replicate the genome. That's the correct one. So gene products that catalyze membrane biosynthesis, you could have said yes or no. You have a 50% chance of getting it. Information, no genes encoding proteins involved in membrane biosynthesis. So that's wrong. And then the other two you didn't pick, which is good. So the answer is enzymes to replicate the viral genome. Let's talk a little bit about the genomes themselves. Viral DNA genomes. Actually, these double-stranded DNA viruses dominate the bacterial virusphere. And the reason I have this here is because RNA viruses do not, interestingly, and we're not sure why that is. And when viruses have DNA, they often emulate the host because our host cells have DNA in them. Many viral DNA are like the host cell, but they're not like cell chromosomes. So our cell chromosomes are assembled, wrapped around chromatin, forming nucleosomes. But most viral genomes are not like that, at least in the virus particle, although when they get into the nucleus, they are what we call chromatinized. And viruses, and that's shown at the bottom there, that's what chromatinized DNA looks like. DNA wrapped around histones, and that is a regulator of not only packaging, but gene expression. But viruses have done different things over the years that differentiate them from cells, and we'll look at that when we talk about viral DNA synthesis. So here are some double-stranded DNA genomes that we're going to talk about. In this course, we focus on a few viruses. We don't teach you about all viruses, and they're listed here. So we have double-stranded DNA from which you can make messenger RNA directly, as I've already told you. And the messenger RNA gives rise to protein, which goes to make new virus particles. And at the same time, you have to reproduce the DNA, of course, to put in new virus particles. It's a simplified view of what's going on. So we have two divisions here, depending on whether the DNA is copied by a host DNA polymerase or by a viral DNA polymerase. So on the left, an example of genomes where the DNA is copied by the host. These are polyomaviruses. They're 5,000 bases of circular double-stranded DNA. They're too small to encode a DNA polymerase. So they just encode some regulatory proteins and, of course, a capsid protein. But on the right are genomes of DNA viruses that are considerably bigger, For example, adenoviruses, 30 to 50,000 bases of double-stranded DNA. Herpes viruses, 120,000 to 240,000. Pox viruses, even bigger, all double-stranded DNA. These viruses encode their own DNA polymerase, presumably giving them flexibility in its reproduction. And so here are examples of viruses with double-stranded DNA genomes, and we will talk about these throughout the first half of the course in terms of how they reproduce. We have adenoviruses, herpes viruses, papillomaviruses, polyoma, and pox viruses. And they're more or less to scale, except the giant virus there, the pithovirus, would not fit on the slide if I made it to scale because it's so much bigger than all the others. And that also has a double-stranded DNA genome and a very unusual structure, which includes what the French have called a cork, that structure, the yellow structure there. And I guess because they like wine, they called it a cork, because apparently this comes out in the cell and out comes the DNA. So they likened it to a cork. So all of these include viruses that infect us, and we'll be talking about those in the course as we go along. Single-stranded DNA genomes are shown here. Remember, a single-stranded DNA cannot be transcribed to make mRNA, so it has to be made double-stranded first. It turns out that viruses with single-stranded DNA genomes can package either the minus or the plus strand. It doesn't matter because when a single-stranded DNA gets into a host cell, this will go in the nucleus, it raises alarms because there shouldn't be any single-stranded DNA in a cell. It turns on a repair system which will rapidly make this single strand into double strand. So the virus DNA, the single-stranded DNA goes in the nucleus, the cell sort of freaks out, repairs it, and now it's double-stranded and it can be transcribed to make mRNA, which of course will give rise to proteins and eventually virus particles. And there are two viruses shown here. On the left, a circovirus, single-stranded circular DNA, rather small. One of them is called TT, which is in all of us as well. All of our blood has this virus. And then on the right, the parvoviruses. These are unusual. They're mostly single-stranded with some unusual T structures at the end. We'll talk about what those do. The B19 parvovirus causes a human disease called fifth disease. There are 5 different rash diseases that kids get, and this is the 5th one discovered. And if you have a dog or a cat, you know that the parvovirus could kill them. So you got to vaccinate them against this virus. Single-stranded DNA genomes. All right, which DNA genome on entry into the cell can be immediately copied into mRNA? This is a nice thought experiment, but it's not too hard. Which DNA gets in can be immediately copied? Double-stranded DNA, gapped double-stranded DNA, circular, single-stranded, linear single-stranded, all of the above. Okay, most of you got double-stranded DNA, which is right. It's the only DNA that can be immediately transcribed without further modification. The gapped has to be repaired. The circular single strand has to be made double strand. The linear single strand has to be made double strand. So all of them need some manipulation before they can be transcribed. RNA genomes. These dominate the eukaryotic virus sphere, and they're rare in bacteria. So this little chart, which is hard to see, shows you 3 different kinds of RNA viruses: plus RNA, double-stranded RNA, and minus RNA. And on the right is a bar graph showing the different families of each in plants, invertebrates, bacteria, archaea, and other, which includes algae, fungi, yeasts, and protozoa. So you can see the plants have a lot of RNA viruses, so do invertebrates, but bacteria and archaea have very few RNA viruses, yet they have lots of double-stranded DNA viruses. Something to do with their evolution, but not quite clear. Now, the key to understanding RNA viruses is that mammalian cells have no RNA-dependent RNA polymerase. That's an enzyme that is needed to copy RNA. Plants and lower animals actually have RDRP, which is the name I give this, RNA-dependent RNA polymerase, but it's used to make interfering RNA, small interfering RNAs. It doesn't reproduce virus genomes. So basically, an RNA virus, in a cell, there's no enzyme that can copy the genome of an RNA virus. And in contrast to DNA viruses, where some viral genomes are copied by cellular DNA polymerase. So the consequence of that is all genomes of RNA viruses, I take that back. Genomes of RNA viruses need to be copied by their own RNA polymerase. Most of them encode in RNA polymerase. There's always a light going on every time I say that. There is one exception that we're going to talk about later. But for the most part, you can assume that the vast majority of RNA virus genomes encode their RNA polymerase, and that is what reproduces the genome and makes mRNA from it as well. The one exception we'll see much later on in the course, but for now, this is what you need to know. All right, so let's go through the different kinds of RNA genomes. First, we'll start with double-stranded RNA. Remember, double-stranded RNA has a plus and a minus strand together. You know, just like DNA, double-stranded DNA plus minus, this has plus minus. And so, by the way, all these figures, which come from mostly from the book, the nucleic acids are color-coded. So the Kelly green, which is the mRNA, that's plus, and then the olive color is the minus. And for DNA, it's like blue, dark blue, and light blue. Dark blue is the plus and light blue is the minus. So you can see double-stranded RNA has plus and minus, but ribosomes can't get to that plus strand to make protein. They can't reach it because it's double-stranded, it's occluded. So in the virus particle, the double-stranded RNA has to be copied to make mRNA. That's just something you're going to have to remember. And then the mRNA, of course, can go on to make proteins, and these proteins can participate in the reproduction of more RNA. So one virus that is an important human pathogen that has double-stranded RNA genomes is called rotavirus. It's an agent of human gastroenteritis. You know what that is, right? Vomiting and diarrhea. Wonderful disease. And we will talk about that later on in the course. And then these rotaviruses are quite interesting. They have segmented double-stranded genomes. The genome is in pieces. So in the virus particle, the genome is in pieces, each encoding a couple of proteins. And you can see them there. They're different sizes. So don't ask why it's in different pieces. You should ask what's the function of having it in different pieces. And it does give you some advantages compared to having one molecule of double-stranded RNA. There's certainly viruses that have a single molecule of double-stranded RNA. I think I said DNA, but I meant RNA. Some of them have 2 segments, some have 4, some of them have 12. Not clear what the advantage is. Then we have plus single-stranded RNA viruses. And these are my favorite because they include the Picornaviridae, which includes poliovirus, which I spent 40 years working on. So I have an affinity for that. These all have plus-stranded genomes, so they can be translated as soon as they get in the cell to make proteins that will go on to reproduce the genome and make new virus particles. And these range in size. You can see the coronaviruses, remember, they can go up to over 40 kb, as we saw on a previous slide. They have 5' and 3' non-translated regions. Some of them have a poly A tail, but you can see the flaviviruses don't have a poly A tail, and they range in size. So these are some of the plus-stranded RNA viruses. And it's just to give you some idea of what pathogens are out there with plus-stranded RNA. So we have the Picornaviruses. So remember, Picornaviridae is the family name. You could also call it the Picornavirus family. Some of the viruses in there include poliovirus, which causes paralysis, right? And rhinovirus, which causes common colds. Caliciviruses are another family that cause gastroenteritis besides the rotavirus. This is another one, vomiting and diarrhea. Then we have our coronaviruses, SARS-CoV-1, MERS coronavirus, SARS-CoV-2. These are now enveloped viruses. They have a membrane on them, whereas the Calici and the Picornas did not. Then we have Flaviviridae, yellow fever virus, West Nile, hepatitis C, Zika virus. These are all members of the Flaviviridae. Flavi, or flavus in Latin, means yellow, because the first virus of this family discovered was yellow fever virus. Why is it called yellow fever virus? Well, it reproduces in your liver. It gives you jaundice, and the whites of your eyes turn yellow. So hepatitis viruses, their effect is very easily seen by looking at the whites of your eyes. And then we have toga viruses, like rubella virus and equine encephalitis virus. So quite a few human pathogens. All right, then moving on to another class in that Baltimore scheme of plus-stranded RNA viruses. These have a DNA intermediate. These are very interesting. Well, they're all interesting, but these are a little bit more perhaps because of what they can do, and we'll talk more about them separately. The family is Retroviridae, or the retrovirus family, and there are a lot of viruses in it, but there are two human pathogens, HIV, there's HIV 1 and 2, and human T lymphotropic virus. They are both retroviruses. Now, these are quite interesting because they have plus RNA in the virus particle, and you might say, oh, is it translated when it gets into the cell? And it's not. That RNA is made into DNA because inside the virus particle, there is an enzyme called reverse transcriptase that copies RNA into DNA. First, it makes a single-stranded DNA, then it makes a double-stranded DNA. And when this was discovered in the 70s, it blew everyone's mind because nobody thought this nucleic acid information would go backwards, right? We thought it went DNA, RNA, protein. And now we have RNA, DNA, and then this DNA, of course, is transcribed to mRNA to make protein. So it's going back a little and then forwards. That DNA, by the way, in the reproduction cycle of these retroviruses, integrates into the chromosomal DNA. And it has to integrate in order for the virus particle to be made. So we will look at that process later on. But these, again, are plus-stranded RNA genomes with 5' and 3' and translated region and poly A tails. Now, minus sense RNA viruses, also medically important. A minus RNA cannot be translated, so it has to be copied first to a plus RNA so it can be translated into protein. And because the cell cannot do this, the cell cannot copy the minus RNA into plus, these viruses have to have in the virus particle the RNA polymerase. So this is different from the plus RNA viruses, with the exception of retroviruses. Plus RNA viruses don't have a polymerase in the host, in the virus, because the plus RNA can be translated in the cell to make the polymerase. But these viruses, the negative strand, cannot be translated, so these viruses have to carry the polymerase in the particle. And here we have viruses with segmented genomes, influenza viruses, famously segmented negative strand genomes, or non-segmented. Measles virus, an example of that. Rabies virus, another one, non-segmented negative strand RNA genomes. Here are some examples of viruses that are pathogenic for humans that have negative stranded genomes. Measles and mumps are paramyxoviruses. Rabies is a rhabdovirus. And you can see very different morphologies. These viruses all have membranes on them. Filoviruses, Ebola virus and Marburg, the very long filamentous viruses, influenza viruses, and then Lassa virus. This is an example of an Ambisense viral genome we'll take a look at in a moment. Now, I have this book here. This is very dear to me, this book. This is Fever by John Fuller, which was written in the early 70s. This is a story of the discovery of Lassa virus in Nigeria. And part of the story takes place at Columbia University Medical Center. But I graduated college and I wasn't sure what I wanted to do with my life. I read this book. I was working in a lab. I read this book and I said, holy crap, that viruses can do this? I have to be a virologist. So this is why I'm talking to you today. This book, you should read it. It's really good. It's an easy read. It's the first emerging virus. It came from a mouse and infected people. It still does it to this day. Thousands of infections a year throughout Africa. Being segmented has an advantage that's clear, and that is called reassortment. What is reassortment? So if two influenza viruses infect a cell, they both have segmented genomes. Those genomes, as they're reproducing, can mix so that the virus particles that are made can have a mixture of both of the parental genomes. So for example, if we infect with a red genome and a blue genome, maybe we'll get progeny with mostly blue and one red segment. That's called reassortment. And it's very high frequency. And it is one of the reasons why influenza viruses are a problem, because they can undergo variation in this way. There is another process of exchange of genetic information called recombination, right, where the strands actually are combinations of the sequences from different parents. It's distinct from this, because here the segments simply move into new virus particles. And finally, Ambisense RNA genomes. They have both plus and minus components. These include arenaviruses and others. And you can see that the light green is the plus part, and then there's a minus part. And these viruses, by the way, have a RNA polymerase in the particle. Now, I've been drawing RNAs as lines, but in virus particles and infected cells, they're not lines. They're highly structured. So here, for example, in part B, this is a plant virus RNA. And RNA can assume these secondary structures called stem loops, where the RNAs base pair with each other. And you can see they can form quite extensive stem loop, complicated stem loop structures. There can be long distance interactions. So this red area, these two red sequences actually base pair and bring the RNAs together. So RNAs are highly structured. And that's shown again on the right. That's a element of the HIV genome with a lot of stem loops. And this is the actual three-dimensional structure of that RNA. So even drawing an RNA in this base paired fashion is not realistic, because it's actually got a three-dimensional aspect to it. All right, what statement about viral RNA genomes is correct? A, plus single-stranded RNA genomes may be translated to make viral protein. B, double-stranded RNA genomes can be directly translated to make protein. C, plus single-stranded RNA virus replication cycles do not require a minus strand intermediate. And D, RNA genomes can be copied by host cell RNA-dependent RNA polymerases, or all of the above. Most of you got A, which is correct. Plus single-stranded RNA genomes may be translated. That's the key there, may be. Not all of them are. Double-stranded RNA can't be directly translated, right? Because the double strand, the minus strand, blocks the ribosomes from getting to the plus strand. Plus, single-stranded virus replication cycles do not require a minus strand intermediate. There was a question that was just asked, do you need a minus intermediate? Yes. In order to make more plus, you have to copy it, make a minus, and then you have to copy it again. So that is not correct to say it's not required. They can be copied by host cell RNA polymerases, no, and it's not all of the above. Let's talk now in the rest of today about how to manipulate the virus genome. Now, many years ago, which means before the 70s, people who wanted to study what a gene did in a virus made mutants. And so you would take a stock of virus, the only way we knew how to make mutants, take a stock of virus and treat it with a mutagen or shine UV light on it, something like that. So now you have a tube of virus full of all different mutants. How do you study them one by one? Well, that's where the plaque assay comes in. You take your virus, you treat it with, say, UV light to make changes, and then you do a plaque assay. So now you can pick individual viruses that have individual mutations in them. So the plaque assay was essential to introduce the world of genetics to viruses, and that's why people wanted to develop plaque assays. So before I go any further, let me do some nomenclature. Changes in nucleic acids and proteins. So a mutation is defined as a change in either DNA or RNA. It can be adding nucleotides, removing them, rearranging them. Any alteration of DNA or RNA is a mutation. And these mutations, of course, lead to amino acid substitutions, additions, or truncations, depending on the change. And as I've said, in the old days, we used to introduce mutations by mutagenesis with chemicals or UV light. It is not correct to say a mutant protein, a mutation in the protein, because a mutation is defined as nucleic acid, okay? It's just the nomenclature. It's just the way it was defined originally, and that's what I do in this course. Since the old days, we're not in the old days anymore, we're in modern era of genetics, we can make mutations in a different way. We have what we call infectious DNA clones. We take a DNA copy of a virus, we can make mutations in it, and then we can put those DNAs into cells and recover mutant viruses. And this is, when you think about it, a modern validation of Hershey Chase, because if you take viral DNA and put it in cells, out will come viruses. So it shows you that the DNA is the genetic material. And of course, if you're talking about an RNA virus, you can get some reverse transcriptase, you can buy it, and you can make a DNA copy of that RNA virus and make changes in that. You can make all kinds of changes, deletions, insertions, substitutions, missense, nonsense, et cetera. And we use this ability to make viral vectors that we use for therapy, gene therapy. We're going to talk about that in the last session of this course, how we have developed gene therapy vectors to correct genetic diseases, to kill cancers, to be vaccines. Really amazing what we can do. And one of the key parts of this is inserting viral DNA into plasmids in bacteria. There's a bacteria with its genome, and then it has these little smaller circular DNA circles called plasmids that we can manipulate. We can insert viral genomes into them, and then we can grow the bacteria, make lots of it, and make our alterations, right? So that's what we do now. We have infectious copies of viral genomes. So let me give you two examples of this. First, here's poliovirus. Poliovirus is an RNA-containing virus plus stranded RNA. Of course, if you take poliovirus and you infect cells in culture, you get an infection, assuming you have the right cells. If you extract the RNA from poliovirus particles, very easy to do. You can make purified viral RNA. You can introduce that RNA into cells by a procedure we call transfection. That will initiate an infection. Because the RNA is plus stranded, it goes in the cell, it's translated, and starts a reproduction cycle. If you take that viral RNA and make a DNA copy with reverse transcriptase and put that DNA copy in a plasmid, so now you can make lots of the plasmid in bacteria, you put that plasmid into cells, it will initiate an infectious cycle. And you can also make RNA from these plasmids in vitro using T7 RNA polymerase, which is used to make the mRNA vaccines, by the way. So I find this funny. We used to do this all the time in the lab. We'd make nanograms of RNA. We were so happy to do that. And of course, we'd make kilograms of mRNA for the vaccines, the mRNA vaccines, different scale. Same process, though. You have a plasmid with the spike gene in it and a promoter, and you do in vitro RNA synthesis. Anyway, you can make RNA transcripts of poliovirus. And we do this now because they are much more infectious than is the DNA. So that allows you to manipulate the viral genome. You can't manipulate RNA. And so a DNA copy gives you the ability to do that. You can do the same thing with influenza virus. You can do this with any virus now, okay? Any virus that you want to work on, you can make a DNA copy and use it to manipulate the genome. You can put genes in it, you can take genes out, and so forth. So for influenza, it's a little trickier because there are 8 segments. So you have to have a plasmid for each segment. And the plasmid is shown here. It's a little complicated. It's a double-stranded DNA with 2 promoters. Remember, RNA polymerase in the cell comes in 3 different varieties, Pol 1, 2, and 3. And this plasmid has Pol 1 and Pol 2 promoters, so that when you put it in a cell, you get both plus RNA, messenger RNA to make proteins, and minus RNA, which goes into virus particles. So if you want to make influenza virus, you take 8 plasmids, each encoded one of the segments, you put them in cells, all 8 together, and out comes influenza virus. It's very cool. So you can manipulate influenza virus in whatever way that you wish. So this is interesting technology. It was used quite a few years ago to recover the 1918 influenza virus. So you all surely know that in 1918, there was a huge pandemic of influenza that killed many, many millions of people. But we didn't have influenza virus until 1933. We missed it. We couldn't get it. We didn't know how to isolate influenza virus from these people. So the pandemic came and went, no influenza virus isolates. And even in 1933, we didn't have the 1918 virus because it was gone by then, right? So what do you do? Well, fast forward to the 1990s. And a number of investigators at NIH had the idea to go into the bodies of people who had died and try and get the viral RNA out and sequence it and make a DNA plasmid encoding it. And so they did that in 2005. So this is the cool part. This 1918 pandemic coincided with World War I. So in the US, many soldiers died of influenza. Their pieces of their lungs were taken out and embedded in formalin and paraffin blocks, and they've been stored in the Army's storage area ever since. So these investigators went and got a few, extracted the RNA, sequenced it, and eventually assembled all 8 plasmids, DNA copies of all 8 of the RNA segments of influenza virus. The other thing they did, because they couldn't get the entire sequence from these little paraffin blocks, they went up to I think somewhere north of in Alaska, I'm not sure exactly where people had been buried and they were frozen ever since 1918, never thawed out. They opened the graves, they did a biopsy of the lung. They got permission to do this, of course. It's not like they went at night and opened the grave, right? And they sequenced the rest of the genome from those corpses in the ground, buried in the permafrost. So anyway, I interviewed Jeff Taubenberger just last year, at the end of the year. He's at the NIH. He's one of the people who did this. And it's a really interesting interview where he talks about this. It took him 10 years to do this because they were using old DNA sequence technologies. So they took these 8 plasmids, they put them in cells, and out comes the 1918 influenza virus. I bet you'd be scared if you read about that, right? Well, most people were scared. They had to do it under BSL-4 conditions, which is the highest biosecurity, which we will talk about later. And many people got upset at this experiment. They said, why are you bringing this back? It could kill us all again. There was a big debate initiated by this work. I mean, I happen to think the benefits outweigh the risk as long as you do the experiments safely, right? But there are many people who do not. Okay, so that's one example of recovering a virus that's no longer around. You get the sequence and then you clone it. But actually, you don't need to clone the DNA all the time. And this is an example of that. So this is Horsepox virus. So Horsepox virus has been around for many years. It's not a human virus. It infects horses, as the name would say. And at some point, it went away. It became extinct. But people had done the genome sequence. So it was decided in several labs that it would be worthwhile to get this virus because it could have some medical uses. So we have the 212,000 base pair genome sequence. And so the investigators had chemically synthesized the genome in 10 overlapping fragments of DNA. So, right, those are the red bars there. Those are the 10 fragments. 150 grand to sequence this. It was expensive. So, you have to hope that it's going to work, right? And if you take those 10 DNA fragments and just throw them into cells, the cells take them up, they recombine, and they make infectious virus. And they actually put some markers in these fragments to show that the virus they got still had those markers and it was what they put in. So you can recover virtually any virus as long as you have the genome sequence. Now, of course, this kind of technology gets people afraid because they're worried that terrorists might, well, smallpox is eradicated, right? But you could find the sequence of smallpox virus in GenBank, and you could make it if you wanted to. It would probably kill you in the process, but maybe somebody wants to do that. So after these experiments, people started to think maybe we should regulate this kind of research. And so here's a headline that was in a journal. Scientists bring back extinct horsepox, raising important biosecurity questions. So then the US put together this advisory board, NSABB. It's a federal advisory board which oversees these kinds of experiments. So if you, say one of you wanted to recover horsepox, you would have to write a proposal and they would look at it and make sure, first of all, that the benefits are worth it and if you're going to do it under the right conditions. And if that's the case, you could do it. But this is a federal agency and they don't have oversight over federal funds like NIH funds. If you're a company, you don't have to fall under these regulations. And so you could do it on your own anyway. This has re-entered the news lately because of SARS-CoV-2 origins. And this brings us to the last thing I want to talk about, and that's gain of function research. You've all heard of this, right? It's in the news every day. Gain of function has become a political tool, which is really terrible. When I started in science, there was no politics, but now it's full of politics. By the way, do you know what happens when you mix science and politics? What do you get? Politics. You get politics. Always. So let's start with the definition. What is gain of function research? Gain of function simply means you give a new property to an organism. Any organism. Bacteria, yeast, virus, plants, animals. But we're going to talk mainly about viruses because this is a virology course. And for viruses, you could do it in the old days before recombinant DNA. You could take a virus and let's say you wanted to get a virus to grow in mice that doesn't usually grow in mice. You could just keep passing it from one mouse to another until you got a variant that did that. That would be a gain of function for that virus. Now, nowadays, you could take the DNA of the virus and modify it in a way to give it a function. So there are two ways to do gain of function. And now we have headlines about gain of function. Risky gain of function studies need greater guidance. And then the COVID origin, some people think they were doing gain of function research and that led to SARS-CoV-2, which is incredibly cockamamie. It's just not, there's no evidence for it whatsoever. And I'd be happy to talk to any of you about that at length, but we can't do it today. NIH documents provide new evidence. We funded gain of function research in Wuhan. Not quite right, but there were interesting experiments going on that I'll tell you about. Republicans spin NIH letter about gain of function research. So now it's become politicized. So it is actually the Republican Party that wants to restrict this kind of research. That's the reason for the confusion. The public thinks gain of function is all bad. So when they hear gain of function, they say, oh, was that where SARS-CoV-2 came from? No. I just say one thing. If you wanted to make SARS-CoV-2 by gain of function, you'd have to have something really close to it. And we have nothing really close to it. Nobody has any virus really close to it. In a 30,000 base genome, 4% difference, which is the closest we have, is 1,200 bases. So that wouldn't work. But anyway, I think the public needs to understand that gain of function can be good, especially if done under the right conditions. Let me give you two examples of gain of function research that have, one of which has been in the news and one not. So on the left is a 1939 paper where they took poliovirus, which doesn't paralyze rodents, and they passaged it in rodents many times until they got a variant that could now infect rodents and cause paralysis. So that's a gain of function. You give the virus a new function. That's the old way of doing it. On the right is a graph from a grant application from EcoHealth Alliance. This is the organization here in New York City that had been collaborating with the Wuhan Institute of Virology. And this is a grant application that was obtained by Freedom of Information Act by reporters who wanted to see if there was something bad going on. So in this experiment, what you do is, the question is, are there viruses in bats, coronaviruses in bats, in caves in China that can infect human cells and potentially be a pandemic threat? Okay, so what you do is you take a coronavirus in the lab, and then you get viral isolate, you get sequences from bats in caves, you get the spike sequence, and you put it into that coronavirus in the lab using this DNA manipulation that I've told you. And this is an experiment where you infect mice, and you ask, do any of these spike genes encode a protein that will make mice sicker? As kind of a measure of, is it a threat or not? And so here what we're doing is looking at body weight loss. That's a good way to measure virulence of a virus in mice, just to see how much weight the mice lose over time. You can see days after infection, percent body weight loss. So the virus you start with is called WIV1. It's in the red. And so you see between 5 and 6 days, the mice lose a little bit of weight. And then you take that virus and you put spikes that you've isolated from bats. These are not actually viruses, they're just sequences. You put the spike sequence in WIV1, you recover virus in cells. You ask, do any of these get more pathogenic in mice? And you can see the green one, is more pathogenic, but some of them actually don't cause any weight loss. There's one up here in the 4231. So that's actually a loss of function. You've taken away the ability to cause weight loss, and others don't really have any difference from WIV1. So this is an example of gain of function. This is the kind of work that Rand Paul was complaining about that we were funding. It has got a goal. It's meant to ask whether there are viruses in bats that could pose a threat to humans. And so, in my view, it's a good goal. The benefits outweigh the risk as long as you do the experiments under the right conditions. In my lab many years ago, here at Columbia, we did a gain of function with mice. So mice cannot be infected with poliovirus. So we took the human receptor for poliovirus. We took the gene that we identified in humans. We made transgenic mice. And now those mice can be infected with polio. They get paralyzed. So we did a gain of function with mice. It doesn't have to be viruses or bacteria. We gave these mice a new property, the ability to be infected with polio. And so I'm going to leave this. You could read this on your own. I'm not going to read it here. But this is an opinion I wrote a number of years ago in PNAS. It was an article describing the kinds of gain of function experiments with coronaviruses that was published. In other words, taking sequences that you obtained from bats and asking, do they encode spike proteins, which could potentially make a human pathogenic virus. And this is kind of an opinion saying that, the people who are critics of gain of function, they say we're going to cause extensive human suffering. And then I go on and explain why gain of function is important. The benefits typically outweigh the risk. Being overly cautious is not without its own risks, as we may not develop the advances needed to identify future pandemic viruses. And so my conclusion is measures should be put in place to allow the safe conduct of these experiments so that the full benefits of science for society can be realized. But this is a huge point of debate. We have even scientists on both sides. We have a number of very vocal scientists who say this is all incredibly dangerous and useless. And then people like me who say this may be dangerous, but it's very useful and we can do them under the right conditions. So the politicians are taking advantage of this debate and trying to regulate these kinds of experiments. All right, so next time we're going to start talking about how to build a virus particle, structure of viruses.